This is our introduction to our final Bayesian project, which is looking at how COVID-19 lockdown impacted animal activity.
Yeah, so this project was inspired by a recent study that was researching the impact the COVID-19 lockdowns had on animal populations, in particular, how animal activity levels changed in response to increases and decreases in human activity. Our main goals for this project are to introduce the mixed effects model and meta-analysis methods, as well as to assess the researchers' frequentist findings using Bayesian techniques.
Absolutely. And during our presentation, we'll also discuss the results, which vary from intuition, where you think lockdown may have benefited animals universally, but that wasn't always the case. Now, the mixed effect model is what we'll be using in addition to a meta-analysis. For mixed effect models, it combines using fixed effects and random effects. This diagram, I think, does a good job of showing how different mixed effect models can function. We're focusing on the top right corner, where we have constant slopes depending on the different characteristics of the animal, and we allow the intercept to vary by species population. So that way we can accommodate changes in behavior will be different at an absolute value across species.
Yeah, so the researchers freely released the data and code they used to perform their analysis. Essentially, they defined a treatment period of higher human activity and a control period of lower human activity and compared the two response variables of amount of animal activity and timing of animal activity between these periods. Due to the scope of this project, we will only be concerned about the amount of animal activity. Under the mixed effects model, there's a set of fixed predictors, some of which are animal traits, the others being human factors. Additionally, there are two random predictors, project ID, and the nested family and species variable, where each species is mapped to the correct family.
Now, we mentioned that our model is going to use a meta-analysis approach, which is similar to a typical linear regression. The difference is a meta-analysis combines data from a multitude of projects. So rather than regressing independent variables on one outcome variable, we're regressing independent variables on the mean of outcome variables from different projects, and then we model the mean by, we model our outcome variable using a normal distribution where the mean is from the regression and the variance comes from the variance of each project.
Yeah, so this is a rough diagram of our hierarchical model where all the beta parameters corresponding to the fixed predictors will be given weakly informative priors. And then for both of the random predictors, we'll sample beta parameters from a normal distribution where the hyperparameters mu and tau are given their own weakly informative priors, and then use the resulting model to sample the data.
And then we'll go into all of this in more detail, but just to give a quick introduction, another unique aspect of our project is introducing how to do a nested random predictor where we have species nested within family. So we generate parameters for each family of animal, and then with each species, we generate, using the family parameters, a new predicted parameter, a beta value that'll act as our random intercept. And this JAGS code also shows the overall model where we have the mean from each project feeding into the normal regression that is then used to generate samples of our outcome variable of interest. We're looking forward to talking to all of you on Monday.